delighted to welcome all the bamboo lovers to today's talk. First of all, I will take this opportunity to thank Bamboo Society of India for giving me this opportunity to deliver this talk and also to share my experiences. In the present scenario, all of us are aware about the importance of bamboo, about how versatile bamboos are with multifarious uses. Hundreds of uses of bamboo have been reported, right from making handicrafts, furniture, scaffolding, flooring, cosmetics, paper industry, jewelry, tissue paper, bioenergy, and food items, and many others. Looking at the importance of bamboo, Government of India has also started many schemes for popularizing bamboo. It has now been removed from the category of the trees, and now no permit is needed if we are growing bamboo on private land outside forest area for harvesting or transporting bamboo. So similarly, National Bamboo Mission has also been constituted in order to popularize bamboo all over India, and it is working in different states. They are providing subsidies to farmers or other growers so that they are encouraged to grow and plant bamboos in their fields and other lands. So there is demand for bamboo planting material for raising these plantations, and these plants can be produced through seeds also. But as we know, bamboos has a unique character after long flowering cycles, maybe after 30 years or 35 years. And sporadic flowering is there in some species, or some species flower gregariously, and some species are not known to flower also. So the supply of seed is not consistent. So one means of producing planting material or plants of bamboo is through vegetative propagation. So vegetative propagation includes two types of propagation, which are macropropagation and micropropagation. Micropropagation includes tissue culture, but today I was asked by Mr. Krishnan to concentrate on macropropagation of bamboos. So concentrating my talk on macropropagation of bamboos. So I will start the presentation now. I welcome you to this presentation, and the title of my presentation is Macropropagation of Commercially Important Bamboo Species. Can I request to go to the next slide, please? So the presentation outline is roughly like this. Bamboo, a wonderglass, its taxonomy, propagation of bamboo, practices of bamboo macropropagation, types of macropropagation, and also macroproliferation of bamboo, and then macropropagation of some important bamboo species. Next slide, please. Just a few general statistics about bamboo. Nowadays, we know bamboo as a wondergrass, poor man's timber, green gold, and it has been known that bamboo grows 3 times faster, and it can be harvested 4 times as often as eucalyptus. And bamboo is found extensively in natural forest and is suitable for deforestation of degraded lands. And some statistics like 2.5 billion people worldwide use bamboo, 1 million people live in bamboo houses, and Indian bamboo sector generates about 432 million work days annually. Next slide, please. And coming to the taxonomy of bamboo, we know that it belongs to the kingdom Plantae, to the family Poaceae and subfamily Bambusoideae. Next slide, please. And some other statistics for all over the world. All over the world, around 90 genera with 1,250 species are reported, and this number keeps on increasing also. And southern China is reported to be richest from the species diversity point of view. And Southeast Asia is known as the continent of bamboo growers. And distribution is from tropical to subtropical region, ranging from 46 degrees north to 57 degrees south latitude. And it grows about the mean sea level altitude from 770 to 1080. And temperature range is from 16 to 38 degrees Celsius, and the grows in rainfall range from 100 to 400 mm of rainfall areas. Next slide, please. And according to the National Bamboo Mission, in India, bamboo covers around 13.966 million hectare of area with 136 species. And according to them, they have given a list of 10 important bamboo species which are more useful for plantations as well as for many end products. And these species are Bambusa tulda, Bambusa bamboos, Bambusa balpua, Bambusa petulacensis, Bambusa polymorpha, Bambusa nutans, Dendrocalamus asper, Dendrocalamus hamiltoni, Thyrocycus oliveri, and Melocanna passifera. Next slide, please. And the bamboo growing areas in India, all of us know that Northeast India is the richest from point of view of the species density as well as the area covered by bamboos. 28% of area of Northeast India is covered by bamboos, which is followed by Madhya Pradesh and then Maharashtra. Madhya Pradesh area is 20.3%, Maharashtra is 9%. And the current usage of bamboo, according to the National Bamboo Mission, maximum bamboo usage is for scaffolding purpose, which is 3.40 metric tons, followed by handicrafts, then paper, and many other uses are also there for the bamboo consumption in India. Next slide, please. And this is again a map of India showing the distribution of bamboo in different localities. Non-clump farming bamboos are 67%, whereas non-clump farming bamboos are only 33% of the area. Next slide, please. This is the map of distribution of map of bamboo distribution throughout the world, and this is for the woody bamboos. That lower map is for the woody bamboos. It's found in almost all the continents except Europe. It has been introduced. Native, it is not found in Europe. Next slide, please. And now coming to our topic, which is propagation of bamboos. As I have already explained, also can be propagated through seeds also by sexual reproduction. But seed is inconsistent in supply. We are not depending on seed. And then when we go for asexual reproduction, that is vegetative propagation, there are many ways for propagating bamboo through vegetative means. That is either through rhizomes or offsets, or we can also propagate bamboos through cuttings. So for cuttings, there are two types of cuttings. The whole culm cutting we can take and make it in pieces, or we can use the branches which are arising out of the clump by making them as culm branch cuttings. And then one process of macroproliferation is dividing the plant into smaller parts. Macroproliferation is there, or you can go for layering also. And the other method is tissue culture, micropropagation, which is now it is being used for mass multiplication of many bamboo species. Next slide, please. So again, this is an overview of the bamboo propagation. And with seeds, we have the limitations, as well as the plants which are produced will be variable because crossing over takes place, and the seedlings which result are not uniform. But through asexual reproduction, that problem is not there. The plants which are produced through vegetative propagation are uniform in nature. So I will be concentrating today on macropropagation. Next slide, please. Different parts of the bamboo plant, like rhizomes, culms, culm branch cuttings, can be efficiently used for propagation, and large scale production of planting stock of different bamboo species can be done. The size, position, age, and season of collection determines the rooting ability of culm cuttings and their survival. There are many factors which are responsible that how much rooting percentage we will be getting when we are using cuttings or culm branch cuttings for production of planting material of bamboos. For that is what is the age of the clump, which is the season in which you are collecting, size of the cuttings, all those things matter for the rooting percentage. Next slide, please. And also, maturation of the donor bamboo culm is also an important factor. Macropropagation tends to preserve desirable traits of the mother or the donor bamboo. So this results in the true to type plants, that is the plants which we are producing will be having the same genetic makeup as the donor bamboo. And macropropagation is easier in juvenile phase than in the mature phase. In the young parts of the bamboo clump, it is easier to get rooting than in the mature phase. And juvenility exists in the bamboo disorder. Rhizomes are the most juvenile part of the bamboo clump, followed by the culms. Then the culm branches are the most mature. These juvenile and the most mature parts are the culm branches. So the rooting will get survival or the plant formation of plants which will be getting will be more in rhizomes, followed by the culm cuttings and the culm branch cuttings because they are oldest or they are least juvenile. These rooting personage will be getting in the culm branches. Success also requires applications of phytohormones and other growth promoters. If we want to have luxuriant rooting percentage, we should be applying some phytohormones like auxins or other growth promoters also to the cuttings. And also the response is different from species to species. Some species will result in good rooting percentage, high rooting percentage, but some species, in spite of addition of the phytohormones also, application of phytohormones also, rooting percentage is less. Next slide, please. So the status of the donor bamboo, the mother plant or the donor bamboo from which we are collecting our material for propagation should be healthy and fast growing. We should not have any insect attacks or any fungus on them. Then if we are doing the collection of propagule at the onset of the active growth phase, that is end of February to end of June in Central India, there is a spring season after the winter temperatures, winter temperatures start increasing. And when the active growth phase starts in the bamboos, that time is suitable for propagation of collection of the planting material, propagules for propagation of the bamboo through cuttings. And this few pictures of different species of bamboos I have shown here. This is the Bambusa bamboos, and this is the tulda, and this is, I think this, I'm not able to see it, it's Bambusa vulgaris. Next slide, please. Next slide, please. Now, coming to the application of rooting hormones, as I told you, for getting luxuriant growth in the cuttings, we need to apply some rooting hormones. Here we use auxins most commonly, and if auxins are not available for nursery personnel and all, they can use the commercial Rootex is available, but we here use auxins. Many kinds of auxins are there, and like indole acetic acid IA, indole butyric acid IBA, naphthalene acetic acid NA. And with our experience, we have found that indole 3-butyric acid IBA is the most suitable auxin for rooting, getting higher percentage of rooting all the bamboo species. And we give the treatment by immersion of the cuttings in practice. But usually what we do is we make the cuttings of the size we require, and then we immerse these cuttings in the solution of these auxins. Next slide, please. And now other important thing, in addition to the auxin treatment, is also the propagation environment. Macropropagation requires stimulation of certain conditions such as 70 to 80% relative humidity and 30 to 35 degrees Celsius temperature. Usually what we do is we propagate our bamboo cuttings, put our bamboo cuttings in mist chambers, which are having high relative humidity as well as temperatures are also maintained here. The micro-free environment is necessary for the success also. The hygienic conditions should be there, free from all kinds of pathogens and microbes. And usually we are using sand beds in the mist chambers. Sand we are putting in the beds in the chamber, and we are using it as a propagating medium. And drainage system should also be adequately maintained. If drainage is not proper in the medium, rotting of the cuttings will take place. And if we are not mist chambers, if mist chambers are not there, there are many economical alternatives also. Like we can use polytunnels, small polytunnels can be constructed, or polyglobules can be constructed at low cost, and they are also easy to move from one place to another. Next slide, please. And these are the environmental conditions for macropropagation. This is the view of a mist chamber. Here, humidity as well as temperature is maintained. Proper sanitation should be there, and protection against fungi and other insect pests also should be taken care of. Next slide, please. And this is again a view of the mist chambers with the beds having sand in them. These the upper two photographs are for the mist chamber, and the lower photographs you can see these are the low cost polytunnels in which we are propagating our bamboo cuttings. And you can see luxuriant sprouting as well as rooting is also very good under this low cost polytunnels, which we have constructed with the use of aluminum frames and this polythene sheets. Next slide, please. This is again the propagation in the mist conditions, sand beds with the cuttings have been planted, and this is the sprouting after around 15 to 1 month. Next slide, please. And now coming to different types of macropropagation, offset planting. Offset planting means, you can see this is the picture of an offset, which is having rhizome as well as some part of the culm is also there with this. The picture, this is known as an offset. And it is the offset is the lower part of a single culm, usually 1 to 2 year old with 3 to 5 nodes with the rhizome axis. Rhizome along with the base of the culm is used for propagation. Planting of these is the most conventional way of propagating bamboo. Offset planting is the most conventional way and it is used commonly. Next slide, please. And so these are a few of pictures of the offset planting. This is a collection of offset planting digging from the mud. These are the offsets which have been dug out, and then this have been planted out in the field. And this we have got the sprouting in the offsets. You can see the sprouting here in the offsets. Next slide, please. Rhizome planting is also one of the methods for propagation of bamboos. And this you can see the rhizome along, part of culm is not there, only the rhizome has been dug out from the soil for propagation purpose. And these rhizomes have been traditionally used to propagate clump forming bamboos. And these rhizomes can be easily dug out in the rainy season and planted in the field. And the new shoot emerge out around 15 to 20 days. Next slide, please. And when we are collecting rhizomes or offsets, they should be collected from healthy clumps, and care should be taken that no damage is there to the rhizomes, and also no damage is there to the roots which are attached to the rhizome. The underground rhizome and roots of the offset should be immediately wrapped in leaves or gunny bags or moist sawdust or straw for transportation. If we are transporting these offsets or rhizome from one place to another, they should be wrapped immediately in moist gunny bags. Transport the offsets to the planting area or nursery as soon as possible, because otherwise some damage will be there and the survival possibility in the field will be less. Next slide, please. But though this is very successful method of bamboo propagation, but there are some limitations of bamboo offset and rhizome propagation. Survival rate of bamboo offset rhizome propagation is almost 100%, but some limitations are there. That is the bulky side is the most difficult part for that. Offsets are bulky and heavy, and therefore extracting, packing, and transportation of these bulky rhizome offsets is time-consuming, labor-intensive, and also expensive. It takes a lot of time to dig out and transport. It's also expensive if you take labor. And availability of offsets in large number is also limited. Only 2 or 3 can be dug out from per clump. If more are excavated, the regeneration of the parent clump can be lost. Some adverse effect can be there to the parent clump if more number of offsets are dug out. So only limited number can be dug out from the parent clump. Damage to the rhizome buds during extraction is there, so that will cause the low rate of survival in the field. Next slide, please. And this is again a representation of the propagation of bamboos through culm propagation. When we are going for propagating bamboos through culms, we can go in 2 or 3 ways. Either we can use the whole culm for the propagation, or you can cut the culms in small cuttings, like for 1 node cutting or 2 node cuttings or 3 node cuttings. You can cut them single node cuttings, 2 node cuttings, 3 node cuttings, and then this are given treatment, immersion treatment with the oxygen solution, and then they are planted in the beds in the mist chamber. So when we are using whole culm cutting, either we can use the whole culm cutting and give the oxygen treatment, or you can use a method known as cavity treatment, which I will deal in detail later on. Next slide, please. When we are using whole culm propagation, the whole culm will be cut from the parent culm, and planted. The branches will be trimmed and the whole culm is planted in the nursery beds. When the rooting occurs, the rooted nodes are cut and transferred to the polybags. And next slide, please. And in addition to whole culm, we can use the culm cuttings, which we commonly follow here in our institute. We are using culm cuttings, either single nodal cuttings or double nodal cuttings. Culm cuttings are segments of the culm or the stem, usually 1 or 2 nodes with buds and branches. The culm selected for the cuttings are 1 to 2 year old with healthy buds. February to April or June is the best time for cuttings. Culm segment cuttings sprout within a week after planting and root between 45 to 90 days. Sprouting takes place very fast, but rooting process is completed in 2.5 to 3 months. Next slide, please. This is the depiction of the vegetative propagation of bamboos. You can see this is a single node cutting in which rooting has been obtained. This is the luxuriant rooting which we get. Next slide, please. When we are using single node culm propagation, the propagation material consists of selected culm split into 1 node cuttings. Here we can use 2 to 3 old culms also from healthy culm and prepare 1 node cutting. The branches attached to the node should be pruned to contain 2 to 3 branch nodes or about a length of 10 to 30 centimeter branch pruning. Care should be observed not to injure the existing buds. Buds are there on the nodes, so we should take care that the nodes are not damaged. The 1 node split culm cutting should be potted or placed as quickly as possible to the propagation bed. Next slide, please. This is again this you can see. This is the donor plant in the field, the bamboo clump which we have selected for propagation purpose. From there, we bring branches or culms and then we cut them into small cuttings. This you can see a single nodal cuttings are there, in which we have given them immersion treatment with IBA, solution of IBA, immersion treatment is there. Then after that, we are planting that in the soil, sand, and this is sprouting. And after the rooting is there, we dig out the cuttings in this way, and this is the rooting in the single node cuttings, and then we transfer them to the polybags. In this way, the plants are reduced. Next slide, please. And similarly, like in single node cutting, we can also use double node cutting. Instead of single node, we can use double node cuttings. And then similarly, we can give them the auxin treatment and follow the plantations similar to the single node cutting. And then one other method is there, that which is the cavity method. Sometimes when we are using double nodal cutting or even 3 nodal cuttings, we can go for cavity method. This cavity method we have found very successful for some species which are otherwise difficult to root, like Bambusa balboa and Bambusa tulda. In this, what we do is in the middle part of the node, we make a cavity. In the internodal region, we are making a cavity with the help of a sharp chisel or drill machine in the internodal region. And then around, we are pouring solutions of 500 to 100 ml solution of IB or NA or IA, whichever auxin is suitable for you in this internodal cavity. And this cavity will be wrapped with time with a polythene strip or tape, and then it is planted in the sand. Next slide, please. This is the depiction of that. We have brought double nodal cuttings. You can see 2 nodes are there. And in the internodal region, we have drilled a cavity here, and with the help of a funnel, we are pouring the auxin solution inside the cavity. And after that, we are closing that cavity with the help of tapes or polythene strips, and then we have planted them in the sand beds, and then we get the sprouting as well as rooting from this cavity method. This is a very successful method we found for Bambusa balboa. Next slide, please. And then coming to the culm branch cuttings, I told you the culm branch cuttings are least juvenile or the most mature part of the bamboo plants. So we get less amount of rooting percentage in this culm branch cuttings, but this is useful because culm branch cuttings we can get in large numbers. Thick-walled species with stout branches are ideal. For example, for bamboo species as well as dendroclamus species, culm branch cuttings can be used. Branches should be collected 2-year-old culms. Cuttings are made by trimming leaves and small branches and branch tip vesicatures. The branch is trimmed to 2 to 6 nodes with healthy buds. The cuttings are placed into bed 2 to 3 centimeters apart. Sprouting is usually seen after 7 to 10 days, but rooting takes a larger period of time. And a new culm will develop from the base of the cutting within 30 to 60 days. Next slide, please. And we are using culm branch cuttings because it is comparatively less economical part of the bamboos, available in large numbers, requisite numbers, and easy to handle because of their small size and light weight. Next slide, please. And this is the thin ones. You can see these are the branch cuttings. Single branch cuttings they are planted here, the sprouting and the rooting. The thin, the small and thin branch cuttings. Next slide, please. And what we can, in addition to the cutting method, we can also do the macropropagation of bamboos through layering. And it is a method of bringing a culm or branch in contact with soil or sand or rooting medium so that propagation occurs. And layering can be of many types. It can be ground layering or air layering. Here you can see the air layering. On the node, we have wrapped some soil or along with moss, and some oxine solution has been there. And we have tied it with polythene sheet, and at the two ends we have tied it with the polythene strips. And if it is successful before the rain season we do, and we get roots after 2 to 3 months from the node. Next slide, please. So air layering is there, ground layering. These are the two commonly followed air layerings, layering methods of layering. Next slide, please. This is the picture you can see. This is the air layering, and this you can see in the ground. It has the culm has been fixed in the soil. Here also, when the nodes which are coming in contact with the soil, their rooting will occur. And air layering is in the air, as the culm is attached to the mother plant itself. It is not being detached from there. And when the rooting occurs, these portions will be cut and treated as new plants. Next slide, please. These are again the methods which we are following of air layering on the nodes. Next slide, please. And sometimes we get this vertical cutting. This type of culm cuttings are there. Culms are the thin culm branches are there at the base of the major mother clump. Sometimes we dig it out and also use it for planting, for vertical planting. And this will develop into roots later on, and we get plants from them. This is not very common. In some species we get it. Next slide, please. After the plants have been produced, hardening of the rooted plant is very much required. So what we do during hardening is, if we do the hardening, that survival will be more. For that, these rooted plantlets are removed carefully from the propagation bed and placed in the polybags containing a mixture of soil, sand, and farmyard manure in a ratio of 1:1:1, with addition of fungicide in suitable concentration. And then we place this polybags under shade houses, which are having shade of 32%, 50% light intensity. Shade is there, and watering. We water this polybags, but as the plants gets established in the polybags, we can reduce the frequency of watering also. And over watering should be avoided because otherwise they will rot. And then new shoots will start emerging 20 to 30 days after the cuttings have been transferred to the polybags. After 1 month, the plants will be shifted gradually to the next slide, please. And this is a table of the adventitious rooting obtained in culm cuttings of various bamboo species. For Bambusa vulgaris variety green, we find that April to May is the best time for collecting the cuttings. And the diameter of the cuttings we used was 1 to 2 centimeters, and the oxygen treatment was 2 to 200 ppm IBA. And for Bambusa vulgaris, we get very good rooting percentage of 85 to 90%. Similarly, for Bambusa tulda also April, and the diameter size cutting is 2 to 3 centimeters is more suitable, and 200 ppm of IBA is better for the rooting of Bambusa tulda. Similarly, for Bambusa newtons also, but for Bambusa balco, we need more oxygen, 1000 ppm of IBA, and it gives a rooting of 65% with a thicker diameter size of 4 to 5 centimeters. Next slide, please. And one more method is there to increase the number of plants. When we have produced plants through either through cuttings or seeds, we are having plants in the polybags. After a growth of around 6 months, the whole polybag will be filled with pillars and rhizomes. So we can divide this we can divide this bamboo plant with many pillars into smaller parts, and each plant can be treated as a single plant. In this way, we can increase the number of plants. Macropropagation is a technique for fast multiplication of plantlets. Rooted plants with rhizomes are exposed by removing soil and separation and planting of individual shoots with rhizome part and root further development, many shoots in a time span of 4 to 6 months. In a year, we can do this macroproliferation for 2 times, so that we can increase, in this way, we can increase the number of bamboo plants. Next slide, please. So now coming to the propagation of some important bamboo species. Next slide, please. Bambusa balcoa, nowadays, Bambusa balcoa is being planted in, preferred by many farmers for plantation in their fields. So the Bambusa balcoa is some description of Bambusa balcoa. Culms range from a height of 12 to 20 meters. The culm diameter is 8 to 15 centimeter. And the appearance of the culm is grayish green, thick walled, young shoots are blackish, green, brown, or orange leaf culm sheath, and leaves are around 15 to 30 centimeter long, 2.5 to 5 centimeter broad. Next slide, please. This species, that is Bambusa balcoa, can be propagated vegetatively by culm cuttings pre-treated with growth promoting substance such as IBA. Cutting from 2 to 3 old culm gave maximum rooting response. And the uses of this Bambusa balcoa, we know the best and strongest species for building purposes, much used for scaffolding, and it is also used for agarbatti sticks and in bamboo wood industry. As I have told you earlier, the cavity method gave us good results with the in Bambusa balcoa for the rooting percentage. Next slide, please. And then coming to the Bambusa bamboo, the thorny bamboo, and it's commonly known as cotton bamboo also, and it is also known as Indian thorny bamboo or spiny bamboo. This is a picture of a clump which we have selected for propagation. The culm's height is 30 meters and diameter is around 15 to 18 centimeters. And the culms are sometimes not straight but are armed with stout curved spines. And the appearance strong, cylindrical, erect, hollow, dark green colored, and also thick walled culms are there. The leaves are 15 to 30 centimeters long and 8 to 15 mm broad, with about 10 leaves in each complements. Next slide, please. Propagation. This is one of the species in which we get seeds, and seeds are collected and they can be sown directly in nursery bed. Sometimes we get the seeds during March to May in central India usually. And then in these seeds we get the germination of around 90 to 100% germination if fresh seeds are sown. But if seeds are not available, then we go for single or double loaded cuttings treated with 200 ppm IBA, which gave us a rooting of around 60 to 70% in the summer months when they are planted horizontally in the mist chambers. Next slide, please. And this is the picture of Bambusa bamboos vegetative propagation. These are the culms which are being extracted from the mature clump, and then we are cutting it into smaller sizes, preparing it into single nodal cuttings or double nodal cuttings, and giving the inversion treatment. And this is the rooting in the bamboos cuttings. This is the planting stock of the Bambusa bamboos. Next slide, please. And there are many uses of Bambusa bamboos, and it is used as a raw material for paper and pulp industry, panel products, bridges, and for ladders. Other uses include scaffolding, rafters, thatching, roofing, basket making. Many uses are there, and the young shoots are edible, and seeds are also sometimes eaten. And the leaves are also used for fodder and thatching and medicine purposes. Next slide, please. Now coming to Bambusa mutans, which is a very important species nowadays. It has also been prioritized by National Bamboo Mission as one of the important species. So this Bambusa nutans, its height is 6 to 15 meter, diameter 5 to 10 centimeter. Then appearance a medium-sized graceful bamboo, loosely clumped, thick walled, straight, green, smooth, not shining, white in below, more slightly thickened, often hairy. Leaves are also there, linear lanceolate, upper surface is dull green. Next slide, please. And we conducted propagation trials with culm and culm plant cuttings, and in this single noded culm cuttings treated with 200 ppm of IBA and planted horizontally during the summer months gave 70 to 80% of rooting. Next slide, please. And this is the propagation of Bambusa nutans. This is the rooting here which we have achieved in the single noded culm cuttings. Next slide, please. And this here you can see that Bambusa nutans we have used single nodal culm cuttings with different diameter sizes. This is the 1 to 2 centimeter diameter size, and this is the 2 to 3 centimeter diameter size culm cuttings. Different size diameter size culm cuttings can be used for propagation. Next slide, please. And it is also a bamboo which is used, and it is one of the bamboo species which is commonly used in the Indian paper industry. The culm is good, strong, straight and used locally for various purposes, mainly as poles. Next slide, please. Then coming to Bambusa tulda, and Bambusa tulda is distinctively identified with a white ring on its nodes. You can see here, white nodes are there on its nodes. So culm height is around 7 to 23 meters, and the culm diameter is 5 to 10 centimeters. And the appearance is a glabrous green when young, gray green on maturity, sometimes streaks with yellow. Nodes are slightly thickened and it is a thin wall. And the leaves are 15 to 25 meter long and 2 to 4 centimeter broad, linear lanceolate. Next slide, please. And single nodal cutting also we have used, giving to a treatment of 200 ppm IBA. And summer season gave the best rooting response, and this is the rooting in the tulda single nodal cuttings. Next slide, please. This is again the pictorial representation of tulda macropropagation to cuttings. Next slide, please. Similarly, here also we have used different diameter classes of rooting in Bambusa tulda, and we get good rooting in the 2 to 3 centimeter size of the diameter of the bamboo cuttings. Next slide, please. And this Bambusa tulda is a very useful species. Nowadays, it is being promoted for agarbatti industry also. And this species is used throughout Northeast India for covering the house and scaffolding. The tender shoots are used for making excellent pickles. It is suitable for the manufacture of wrapping and printing paper. And this is one of the 5 most quick-growing species of bamboos preferred for raising plantations in India. It can be used as reinforcement in cement concrete. It is one of the 2 most important edible species, and as I told you, it is being promoted for nowadays for agarbatti industry also. Next slide, please. Now coming to Bambusa vulgaris. In our field trials which we are conducting, which we have initiated 2 to 3 years back, we are getting very good growth in Bambusa vulgaris in central India. So this Bambusa vulgaris is also a very promising species for taking up in plantations. Culm height is 8 to 20 meter, diameter is 5 to 10 centimeter. Appearance, it is a bright green, glossy, erect, mature culms, yellowish. Walls are 5 to 15 mm thick, branching, usually covered with brown hair. Young shoots dark brown to yellowish green. The leaves are 15 to 25 centimeter long and 2 to 4 centimeter broad, rounded at the base into 5 mm long petiole. Glabrous on both sides. Next slide, please. And here also, single or double nodal cutting has given us a good rooting response in the summer months, and they have been treated with 100 or 200 ppm IBA overnight treatment. Next slide, please. And this is also a species which is used for paper making, scaffolding, construction, poles, curios, and handicrafts. And it's also commonly used for fencing in Manipur. And the longevity of the culm is comparable to that of the best timber. In Sri Lanka, it is most widely used. It's the most widely used bamboo. It's occasionally cultivated in Malaysia for its edible shoots. Next slide, please. Now coming to the most commonly distributed bamboo species, is Dendrocalamus strictus, and uh, this is most commonly distributed in Central Indian states also. And the culm is height is to 8 to 16 meter, and diameter is thinner, 2.5 to 8 centimeters. Appearance, which is a deciduous, densely tufted bamboo, pale blue green when young, dull green or yellow on maturity, much curved above half of its height, thick walled, nodes somewhat swollen, and leaves are 25 centimeter long, 3 centimeter broad in moist areas, smaller size of leaves in dry localities. Next slide, please. And Dendrocalamus strictus is a difficult to root species to vegetative propagation, and we get lesser rooting percentage through culm and culm branch cuttings in Dendrocalamus strictus. And the use of Dendrocalamus strictus are, it is used extensively as a raw material in paper mills and also for a variety of purposes such as construction, agricultural implements, musical instruments, furniture, etc. Young shoots are commonly used as food. The portion of the leaves and nodes are siliceous matter is used in the traditional medicine. It is found suitable for accumulation of ravine areas also. This species is one of the two most important species for plantation purposes. Next slide, please. And lower pictures are for the strictus, rooting of the culm cuttings in strictus. This ones. Next slide, please. And this is the vegetative propagation of bamboo through cuttings, collection, treatment, and this is the immersion treatment overnight, and then planting, and then after planting, sampling we are doing, rooted plants we are transferring to the pollinators. This is the whole picture, all of the steps involved in the propagation of bamboos through culm cuttings. Next slide, please. This is again, we have shown the pictures of this chamber in which we have planted the cuttings of different species, and these are the sprouted and rooted culm cuttings under mist conditions. Next slide, please. And we have planting stock of different bamboo species: Bambusa nigrans, Bambusa vulgaris, Bambusa balcoa, Bambusa tulda with us, and we sell them to any stakeholders who come to us for purchase at around 25 rupees per plant, and the forest departments as well as farmers take this planting material from us for plantations. Next slide, please. This is again the view of the planting stock of different species of bamboo which have been raised through culm cuttings. Next slide, please. And here at TFRI Jabalpur, we are having a germplasm bank in which we have done the collection of 4 species of bamboos from 3 different three central Indian states of Maharashtra, Odisha, and Madhya Pradesh. And we are maintaining these different expressions in our germplasm bank for Bambusa bamboos, Dendrocalamus strictus, Bambusa tulda, Bambusa vulgaris, and we are continuously adding them. With Bambusa balcoa also we have started germplasm collection, and we use the propagating material from these germplasm banks, this superior clumps which we have collected from different locations, and do the propagation of our planting material. Next slide, please. And from the plants produced by us has been sold to many agencies, but major under license agreement we have sold bamboo plants of Bambusa bamboos and Bambusa tulda to Telangana Bamboo Mission as well as to the Gujarat Forest Department, and they have shown us send us the pictures that this is the picture of growth of around 6 months. They say that growth is very good and they are growing very good with large number of culms being produced, as well as gaining height and as well as the culm diameter. Next slide, please. This is again the pictures of the plants which we supply to the Telangana Bamboo Mission. Next slide, please. And we also at TFRI we also do propagation of bamboos in tissue culture. There are a few of the pictures of tissue culture bamboo. Next slide, please. This is again the view of our tissue culture lab with the cultures of bamboo. Next slide, please. And this is the cycle of the process of production of plants of tissue culture. These are the buds we use. That is the shoot multiplication and rooting, and then hardening of the plants and production of plants of bamboo through tissue culture. Next slide, please. And again, this is the rooting. You can see in Bambusa tulla, these are the rooted plants of Bambusa tulla raised through process of tissue culture. Next slide, please. Similarly, again, multiple shoot cultures of Bambusa tulla cultures. Next slide, please. Again, you can see this is the plantlets which are ready for hardening and then transfer to the polybags of tulla. Next slide, please. And then we have raised a comparative field trial of the tissue culture raised bamboo of Bambusa nitons, Bambusa tulla, Dendroclamus asper at TFI Jabalpur. Next slide, please. And this is the view of our comparative field trial of tissue culture raised bamboo. Next slide, please. And this is the view of our germplasm bank with the accessions from 3 different states. Next slide, please. And also we are in the process of establishing a bamboo system. At present, we have 46 species of bamboo collected from Northeast India, Odisha, Maharashtra, Chhattisgarh, and Madhya Pradesh. And we are constantly upgrading our collections in the bamboo system. And we hope to add collections of Kerala to our bamboo system in the next 2 months. And next slide, please. And we also undertake training programs for farmers, artisans, and the other stakeholders under different training programs funded by the government, like GSDP Green Skill Development Program, or the Bamboo Technical Support Group programs on nursery management, bamboo nursery and management, value addition, bamboo technologies, propagation and management of bamboos. On these topics, we have been conducting trainings for different stakeholders. Next slide, please. And these are the picture of some of the trainings conducted and during the training pictures during the trainings. Next slide, please. The trainees are taken to the field, our plantations, germplasm banks, and collection of the material is shown to them, and also planting through tissue culture, as propagation through tissue culture, as well as macropropagation. Next slide, please. This is the training being shown the method of macropropagation through cuttings. Next slide, please. And also for value addition, we have hired some master trainers and given the training for value addition and production of some products of bamboo. Next slide, please. And some of the products which we have produced during the training programs. Next slide, please. Same products. Next slide, please. These are some of the products which were produced during the training program. Thank you so much. This is all from my side. And thank you for your patience hearing. Thank you so much.
Thank you, Doctor. That was highly informative and detailed presentation. Ladies and gentlemen, if you have any few questions, please raise your hand and we'll take it one after another. Any questions from the audience? Yes. Hi, Ram. Please go ahead.
Thank you, sir. Hi, ma'am. Ram here. Yeah, excellent presentation, ma'am. Thanks for sharing that knowledge. Ma'am, when you do a column cutting, you use a solution, right? Some kind of solution to propagate. So can you elaborate what kind of solution or?
Yes, I can elaborate. Like I told you, we are using auxins. Auxins. IB, indole glutamic acid we are using, and we are giving it in solution of 200 ppm. So to prepare it, what we do is we will dissolve 200 mg of BA in 1 liter of water. So that will give us a solution of 200 ppm of IBA. And this IBA is not dissolved directly in water. It has to be dissolved in chemical like alcohol. Some alcohol. It has to be dissolved in 10 ml of alcohol. And then when it is dissolved in the alcohol, you can add 1 liter of water to it. So it will make a solution of 200 ppm of IBA. In that, depending on the amount of cuttings you have, maybe you have to prepare 100 liter of solution with the cuttings of 50 liters. So accordingly, you can formulate your measurement of how much quantity of IBA you need, and in the tubs or in buckets, big buckets, you can prepare the solution and you can dip these cuttings in this solution and leave them for around 12 hours. Overnight treatment begin. The next day we do the planting.
Okay. So the proposition varies, depends on the soil or depends on the number of poles, ma'am?
No. It is not related to soil at all. It depends on the species. Here,
Species.
Species. It depends on the species, what kind of species we need, what kind of auxin. Here we have found that for most of the species, IBA is good enough. It's the most effective one, IBA. Auxin IBA is the best auxin we found for most of the species of bamboo. So you can try IBA for most of the species. Okay.
Thank you. Thank you.
Thank you, Ram. Next question from Dr. Vidya. Please go ahead, Dr. Vidya.
Good morning, everyone. Ma'am, it was a very good presentation. I would like to know one thing is that, there is any difference between, very distinctive difference between the thick-walled and thin-walled bamboo for all these procedures, for this vegetative propagation methods?
Yes, it's a good question. This process of culm cuttings is more successful for thick-walled bamboos. Thick-walled bamboos means the bamboos belonging to the Bambusa and Dendrocalamus. Most of the bamboo species, what I described, Bambusa bamboos, Bambusa balcoa, Bambusa vulgaris, Bambusa tulda, Bambusa newtons, are thick-walled bamboos, and this process of culm cuttings is successful for them. For the thin-walled bamboos, like the which are found in Northeast India, like Melocana bacifera or Schizosticum dulva, this culm cuttings is not very successful. Most of the thick-walled bamboos, this process is successful.
Which procedure is best for that species, ma'am? Thin-walled?
Thin-walled, I think you will have more successful will be the rhizome and offset method. And if you are getting seeds, then you can go through seeds. Otherwise, rhizome and offset method will be the best method. Or if you are having a tissue culture lab, that is a very good option for going for mass multiplication of any species.
Okay. Thank you, ma'am.
Thank you.
Ma'am, question from my side. What is the cost of say propagation between macro as well as micro?
Here we are selling macropropagate plants for ₹25, and we have also done the calculation for tissue culture raised plants. It also comes to around ₹30 per plant.
So not much of a difference.
Not much of a difference because because we get more number of plants from tissue culture, that is offsetting the cost. Okay. And in macropropagation material, we get less, labor is more, and that balances it out. As we get more, depends on the quantity of the plants you are able to produce in the tissue culture lab. If you are producing more number of plants, the cost gets reduced.
Okay. Okay. Next question. Let us say we
Hello.
have.
Hello, sir.
Mr. Rajshekhar, please go ahead, sir.
Yes. Madam, good morning.
Good morning.
Madam.
Good morning.
Madam, for single node cutting,
Yes.
how old bamboo has to be selected?
We prefer to take 2 to 3 year old culms.
2 to 3 years old.
2 to 3 year old culms.
And that we can cut it into single node, and we can dip in the IBA.
Yes.
How many hours we have to dip it?
We usually give in the evening and leave it overnight, and the next morning we plant it. 12 hours treatment. Good.
Just like a sugar cane, we can bury in the soil or some containers, and we can get the seedlings, no?
Yes. We can get the sprouting and rooting.
Pardon? Pardon?
Yes. Similarly, we can get the sprouting and rooting. Yeah.
Okay. How many days it is required to planting?
We get the whole rooting process completes between 2 to 3 months. That depends on the species. Some species rooting process will be completed in 2 months. Sometimes it will take 3 months.
Okay. That means after 2 to 3 or 4 months, we can plant it.
That is, we can get the plants. After keeping them in polybags for 1 or 2 more months, we can plant it.
Okay. For planting, how much height that sampling has to be?
2 feet maybe.
2-3 feet.
2 feet. 2 feet. That should be.
Okay. Okay, madam. Thank you. Thank you. Very nice program, madam.
Thank you. Thank you so much.
Thank you. Thank you, Mr. Rashikrapa.
Thank you, sir. Thank you.
Vishwanath Giriraj, sir, please go ahead with your question, sir.
Yeah. First, I am Giriraj, CEO Maharashtra Bamboo Promotion Foundation. Please accept my congratulations for a very good presentation.
Thank you, sir. Thank you so much.
My question is like this: in Maharashtra, ours is a government promoted agency,
Right.
Government of Maharashtra and Tata Trust. So one is, of course, tissue culture, getting from tissue culture.
Right.
There are some issues on that, and then for small numbers, for farmers, tissue culture all the way transporting becomes unviable. Maharashtra being as big state as Madhya Pradesh. So suppose we want to encourage, even on trial basis, small nurseries with locally desirable species.
Right.
What would be the using this macro propagation methods? What would be the scale of investment for a small entrepreneur? I mean, has it been studied? I mean, what type? I mean, I'm not asking a very precise figure, but we need such type of entrepreneurs because little bit of bamboos have to be grown, good quality. Suppose one is in Vidarbha, he cannot get bamboo, for 10 or 15 bamboo, he requires the farm end. He cannot bring it from a, very distant lab, this approved tissue culture suppliers. So just like, for household needs, people buy adha kilo, pav kilo from a shop nearby. They cannot go to the wholesaler and buy it. So what would be the cost? Suppose we want to train, 5-10 people in Maharashtra running these nurseries. There's a lot of technical knowledge required. I have 2 questions. How much training, or whether your center can give, or any place for 2-3 months, young people can be exposed at our cost, A, and what would be the scale of investment to become entrepreneurs?
Yes, I agree with your concerns, sir. For farmers or some other stakeholders to start at a small scale, it will be difficult to transport from longer distances or to go for tissue culture lab and all. I don't have much experience about the financial parts, but I think minimum investment of 50,000 will be needed. I think 50,000 to 1 lakh for at least having some shade house like thing or a like thing to plant the cuttings. So some minimum investment will be needed. Around 50,000 should be enough, I think, for constructing a small shade house. Even with sprinklers, if sprinklers are added and a small shade house is constructed, in that they can plant the cuttings. In Vidarbha area also, I have seen farmers are doing on small scale. They have nurseries on small scale with shade house like structures, and they're growing the cuttings in the soil beds. I've seen them doing that. And for training, sir, you're most welcome to send stakeholders to us. We can train them for 1 month or 2 months. We can make different kind of modules for them. Only 15 days back, we had exposure visits of stakeholders from Northeast India as well as Maharashtra and Chhattisgarh from the MSME Foundation, sir. They brought them.
Which foundation?
MSME Foundation, sir. MSME.
Okay.
They brought us for the exposure visit. And that was for a short time, but we can do trainings on longer duration also. We'll be happy to train more and more people so that they can take up their own nurseries for propagation purposes.
So, Giriraj sir, I will share ma'am's number after this session. You can take it to the elaboratively after this.
Excuse me. Hi.
Giriraj sir?
Yeah,
mine is over.
Myself Baswaraj Beli. I'm from Gulbarga, Karnataka. So this is one of the encouraging session from the madam. So those who are into the bamboo, like recently joining. Okay. So I also felt it's a good. So I just have a few questions. Like one of the question is like, does a bamboo require a pruning? And if yes, how often we need to do the pruning? This is one of the question. And second thing is, like say I'm from Karnataka, Gulbarga. So Jabalpur is like very far away from my place. And if I want to do a plantation of about 100 trees of different varieties, so for me, like sir said, Vishwanath sir said, so for me, the cost is very high. Like it is almost comes to 40,000 to 50,000 for transportation only. And my plantation charges are, my plants charges are around 20 to 25,000. So I'm finding it actually difficult. I'm very much interested in planting in my farmland, but because of the transportation charges and all, I'm stepping back. So how this bamboo society is working on this part?
Okay. Sir, what you can do is identify your nearest KVK, Kendriya Krishi Vigyana Kendra, right? Identify your KVK. Every KVK has got a certain amount of bamboo plants. Next step, if you can't find in your KVK, find out your nearest DFO, RFO, range forest officers. Okay, they will be able to give you a certain amount of plants, but it depends on what is available with us. We will not be able to get whatever is available, what you want. We'll have to probably make do with what is available there. However, if you can spend a trip to Bangalore, I would recommend you to come down to IWST, Indian Wood Science and Technology. Okay, and IWST has a nursery where you can buy this plants. And if you can reach from Bangalore to Hosur, you can also meet Dr. Bharati sir from Bromore Biotech. They have some of the, I'd say 6 to 7 varieties of tissue culture plants. Okay. So these are the options for you.
Yeah. Regarding pruning, ma'am, like how often we need to do if pruning is needed?
We start pruning three years after the plantation, and pruning because we have just started our plantations. Not able to give you from my experience, but maybe at a gap of 2 years, I can get back to you on this question after consulting others who have been in the area of plantation for longer terms. I can get back to you. You can give me your email ID.
Sure, ma'am. I will just post it in a note, ma'am, chat, madam, over here.
Please do. Please do.
Yeah. Thank you, ma'am. Thank you, sir.
Thank you. Thank you. Any more questions from the audience? Great. So,
hello. Yeah, Chandraprabha here, sir. Hello.
Yes, sir. Please go ahead, sir. Chandraprabha.
Yes, sir. I got a small requirement. What is the rate of growth difference between a polybag seedling and a tissue culture plant? What is the because costing wise production of seedlings almost both are roughly similar production cost. Whether it has got an impact on the rate of growth, I would like to know.
With our experience with other species also comparing tissue cultured and seedling growth, in the initial stages, tissue culture plants grow faster, but later on, both the growth of the seedling as well as tissue culture plants is balanced out. There is no significant difference in the growth of tissue culture plant as well as seedlings after a few years of growth, maybe after 3-4 years, the growth rate becomes same. Initially, the plants picks up faster.
Is it a disease resistant, madam? Tissue culture plant, is it a disease resistant?
The plants which are come out from the tissue culture lab are free from microbes and all the fungus and bacteria, but as they get outside, in the outside environmental conditions, you can't have control over the plant. No? So the plant which is going in the field, when it comes out of the lab, you can say it is free from everything. But as it is kept outside environment will affect it. So you can't say that it is disease resistant or anything. We are not giving any disease resistant property under tissue culture. Under tissue culture, we are only propagating it. We are selecting good genotypes and we are propagating those types in large numbers in tissue culture.
Okay, thank you, ma'am.
Ma'am, one more question. Ma'am, in my place, like Karnataka, Gulbarga, I have visited a few farmers who have planted the Bhima bamboo, which is Balkova. So I have seen they are the tissue culture plants. I have seen in a 6 to 8 months of flowering in the plants. Like if I see a row of 50 plants, I see 4 to 5 plants have been flowered at the age of 7 to 8 months. So what is your comment on this? And one more question is, which is the best way? Should we use tissue culture is the best one, or the one which is made out of seed, or one which is made out of the other method that you told? That is the best one. Can you suggest on this, ma'am?
This flowering of bamboo we are coming across again and again, and many factors are responsible for that flowering of the bamboos. Many when they are going in the field, so many stress conditions are there for them. So that stress condition can be expressed as flowering also. So it need not be only the process is responsible, the tissue culture is responsible. Even vegetatively propagated from current cuttings also, sometimes they can flower if some stress conditions are there in the field in which they are growing. Many factors are responsible for that. Huh? If the clump is not dying, if the clump is not dying, then it is okay. Because the seeds are sterile. It's not, they're not setting seeds. So the plant will survive. So many factors are responsible for flowering. And we can't say about seeds, no, because seeds are not available for all the species. So alternative left is vegetative propagation only. For seeds, very few species we are getting seeds. Also seeds we are not getting in sufficient quantity also. So vegetative propagated method is the most suitable option only to go through.
Thank you, ma'am. That was really informative.
Thank you, ma'am.
Also, Mr. Baswa, what we can do is, if you can tabulate what is the kind of environment that you are in, okay, under what conditions you observe this, what is the moisture level in the soil, how often do you water, okay, what is the amount of water that you give to this plant. If you can write down some details, that will also help you to narrow down the problem. Stress can be due to various reasons, and it is difficult for somebody to give you a telephonic or a Zoom session advice on this, right? So what I suggest, you get into the macro details on the field, okay? What is the steps that you're following? Second thing also, your soil profile matters a lot, right? It is not just the planting conditions. Soil profile, identify what is your NPK contents, what is your micro and macro nutrients that are available in the soil. Okay, also identify what is your organic carbon content in the soil. So a lot of work needs to be done rather than say, or rather than what you have observed, I would say, due to which the flowering happens. Flowering, as ma'am said, can happen both in tissue culture as well as in the macro cuttings, right? So you need to identify a little bit deeper work needs to be done on this. My suggestion is try to incorporate natural farming methods. Don't get into chemical farming methods. Don't get into organic farming, right? Look into the natural farming methods. In fact, we have a session which was done by Dr. Vivek. The video is available in the library. And you can, if you go to the video, you'll see what steps you need to do in terms of preparing the soil for a plantation. Just using your homegrown material around your farm. No need to get any chemical fertilizers for this, right? So, if you need any help later, ping me, contact me at the PSA office. I will give you a link. We have a complete presentation and all the entire guide for natural farming techniques. Ma'am, thank you.
Thank you,
sir. Thank you, sir.
We have one more question.
Here, sir.
Mr. Umesh.
Hello.
Yeah, yes, Mr.
Yes, sir.
Please go ahead, sir.
Hello.
Please
Hello.
go ahead, sir.
Yes, sir. Yes, sir. There is a small suggestion from my side to BSI, sir. All
Please.
this every week you are conducting series of cultivation, harvesting, and so many other products related things. Why don't you just publish all this series number 1 to whatever as on today, in a published form, in a booklet, or so that we can purchase, so that it will be hand-on for many other things. I think that will be a good.
We will try, sir, but unfortunately, this is a video series. Translating this into a text needs a lot of time and resources. We would be happy if you have someone who can help us out on this. But nevertheless, the whole content is available on a Bamboo webinar channel on YouTube. So you can revisit this any number of times that you want. In fact, that will also help you because it's a video, it's an AV, audio video. You'll also be able to see what was shown at that point of context. Your suggestion is valid. We will definitely look into this.
But hard copy definitely would help more rather than the video version.
I agree.
Anyway,
we
thank
will
you.
request our speakers to give some information. Ma'am, if you can submit us any guide, that will be helpful. We can publish it in.
Yes, sir. That is the one request. Yes, yeah.
Definitely.
So that those who are the resource person, they can have 2-3 pages notes,
Sure.
so that it can be published. It's their contact number and whatnot. Yes.
Sure, sir. Sure.
Yeah.
Thank you, sir. Thank you.
Thank you.
We move on to our next question from Mr. Umesh. Mr. Umesh, please go ahead.
Hi, sir. Umesh here from Maharashtra. Actually, my suggestion is to BSI. Can you please prepare one or two webinar series for after planting to harvesting things, what to do and what to not be do. Like one our listener or one that he is asking for cleaning pruning, how often it has to be cleaning pruning, when soil mounding is there, what is the micronutrients to be given to the plants, which species, it means maintenance of the plants. Okay, so this series can be arranged by BSI. It is my request for farmers also, because everyone is saying that only planting bamboo, but how to maintain the bamboo? It has to be there. Okay. Second thing is, like rightly said by Giriraj sir, there should be small nurseries in pockets of some areas around 50 kilometers or 100 kilometers surrounding that. But how we listing our nurseries from? Is there any institute for register the nursery for applying or give the certification? This is nursery is supplying such much seeds, nursery certification, like your what you can say, horticulture nurseries, right? Can we give the certifications to this nursery as a forest plant or bamboo plant nurseries? Is there any a channel to register those nurseries in Maharashtra? Please guide us.
Yes, one at a time. Let me answer the first question. Ma'am will answer the second one. Your point about the maintenance of bamboo is very valid. What we'll do is we will try to assemble all the FAQs from various sessions about maintenance and create a guide for you. That will be published shortly. Okay? Now, regarding the rest, the acquisition, I would like ma'am to report it. Ma'am, please.
MP State Bamboo Mission had, I have experience with MP State Bamboo Mission. So they had a scheme that they selected applications are invited from different nurseries as well as tissue culture labs, and then teams were sent for their accreditation, and if they passed, they were accredited. They were accredited, and they were suitable for the farmers to buy plants at a fixed rate from them, tissue culture labs as well as nurseries. But a list of this nurseries and labs for Madhya Pradesh must be available with the Bamboo Mission, but I don't know about other states. The team used to go and visit and see the formats were there. Formats were given, marks for different kinds of natural conditions as well as lab conditions.
Ma'am, apart from, ma'am, excuse me, sorry for interruption. Apart from Bamboo Mission, is there any channel to register this nursery? Because Bamboo Mission have lots of guidelines which everyone cannot fulfill, at least from last 3 years. Okay, so is there any small nursery? If I want to make a small 10,000 plants, okay. So what is the smallest channel to get the nursery aggregated from this forest department or agriculture department like this one?
You can,
Because
you're from Maharashtra.
because there is a lot of schemes for bamboo in Maharashtra, like Pokhara scheme, Manrega scheme. We are not, most of the farmers will not interested in bamboo mission or Atal Bamboo Samruddhi Yojana, whatever. But they want to plant some bamboos in agriculture field also, agriculture schemes also. They need to aggregate from someone else.
So, but
Apart from the bamboo mission.
you want to grow for yourself, or you want to sell the plants, make the plants and sell? Why you need accreditation to nursery?
Because of what happened, ma'am? The in schemes, farmers has been asked for a bill, okay, and there is a approved nursery bill in every scheme. If some entrepreneur starts nursery in small scale, if he wants to start the nursery in small scale, like forestry plants, your horticulture plant, that model, this nursery has to be implemented, and then it is easy to plant bamboos in large scale. This is my suggestion.
You are in Maharashtra, you can explore there. State government
In Jalgaon.
The state government is having some scheme. I'm not aware of the scheme from Maharashtra. You can explore there from the state government. Maybe some schemes must be there.
Okay.
So, Mr. Umesh, thank you very much. Any more questions?
Thank you, ma'am.
Thank you so much.
Any more questions? Okay, so no more questions. Ma'am, that is really helpful. Thank you very much for spending some time with us. On behalf of entire audience as well as Bamboo Society of India, I would like to thank you wholeheartedly for sparing your time and spending this with us and enlightening us.
Thank you so much for giving me the opportunity.
Thank you, ma'am. Thank you.
It was a pleasure. It was a pleasure interacting and also delivering my talk. And
thank you.
It was a very fruitful, interactive session. Thank you so much.
Thank you, ma'am. Thank you. Ladies and gentlemen, we come to the end of today's session, and we will be back next week. Looking forward to interact with all of you. Have a wonderful weekend.
Thank you. Same to you.
Thank you.
Thank you all.
Thank you.
Thank you all.
Thank you very much, madam, and very nice program.
Thank you so much.
Thank you, sir. Thank you.
Thank you.